Well, it's a pleasure to be here, and my talk will have two major parts. One is talking about how we thought about getting to therapies for spinocerebellar ataxias, which then led us also to developing therapies for ALS. And in the last 10 minutes, I'll talk a little bit about genetic testing and some guide points I think that we all need to know about. These are my disclosures. NIH has been good to me, and what is relevant for this talk is we have a very close collaboration with Ionis Pharmaceuticals and Carlsbad to develop antisense oligonucleotides. So this was the outline. Let me start with this. So about now 25 years ago, I was called about a family that had an ataxia. This is a middle-aged man who you see up there, age onset of 29. And his classic cerebellar signs, he walks a bit unsteadily and then has a typical cerebellar sign, he steps out. This is a young man, and I'm asking him not to do pursuits, but fast saccadic eye movements. That's all he can generate. He had an onset at age 5. The matriarch of the family, about age 65, so she has had the disease for 35 years now, classic appendicular ataxia, she can't walk anymore. She's severely impaired. And the next video will show you a young man with the age of onset at 9. He's now 16, already quite ataxic. And what is a bit hard to see, I'm asking him to do a Romberg, and he actually sways more, telling you already that this is beyond the cerebellum and has long tract signs as well. So what we didn't know that this would become SCA2, spinocerebellar ataxia type 2. It followed the identification of SCA1 that was identified in 1993. But it was clear early on that this was more than just Purkinje cell disease, because as I just showed you, patients had slow saccades, some had spasticity, some had neuropathy, some had Parkinsonian signs, and if you look carefully, some patients already had fasciculations. So gives you a hint that this is much more than just a cerebellar disease. In 1996, we were successful in identifying the gene. It was in the early days of trinucleotide repeat genes. We found a gene of unknown function that in normal individuals had 22 repeats, and every patient had 33 or more repeats. And when you plotted the length of the repeat to age of onset, you saw a clear inverse correlation. The longer the repeat, the earlier the onset. So how do you think about therapy? And I think this is an important fundamental slide. You have to ask yourself, what does the mutation that we identified actually do to an organism? So there is most recessive diseases that's relatively easy to answer. Recessive mutations lead to loss of function alleles. Either you don't make the protein at all, or you make a shortened protein, or a protein with a missense mutation that doesn't fold right and doesn't have function. But with dominant mutations, it's a lot more complicated. There are three ways this mutation could act. One is that it can be a dominant gain of function, so the protein just does whatever it does more. It can acquire a new function or a toxic function, but it could also be dominant negative. And dominant negative means that the mutated form of the protein interacts with the wild type form of the protein, but makes the wild type inactive. So you, in the end, have a loss of function. And there could be a dosage response. If you lose one copy, you only have 50%, you get disease. Good example is hereditary predisposition to pressure palsies, where you have a reduction of gene dosage to 50%. So the way you think about treatment is also different. If you have a dominant gain of function, you want to reduce expression. And ideally, you would like to really target the mutant allele. And I will later show you that actually for ASO therapies, and for those of you who are in Huntington's disease know it, the first phase 1 trial was just done. The idea of targeting only mutant huntingtin didn't work, and both alleles are targeted, the wild type and the mutant. If you have a dominant negative effect, you actually do need to target the mutant allele, because if you target both, you just make the situation even worse. If you have a dosage reduction, hemizygosity as it's called, you want to increase expression. And for recessive diseases, you would like to increase expression. That's where really gene therapy type of therapies are coming in. This may seem easy on a slide to sort out. It's actually not that easy, and it could also be that mutations have multiple functions, leading to gain of function in one pathway and partial loss of function in the other one. One way to approach this is to use mouse models. If the disease is caused by loss of function, then if you knock the gene out in the mouse, you should see the phenotype. On the other hand, if it's a dominant gain of function or gain of toxic function, then if you express the mutant form of the protein as a transgenic model, you should see the disease. And then what would you like to see in your mouse model? And for those of you who do Alzheimer's disease, you know it is actually, or Parkinson's disease, it's actually difficult to produce these phenotypes in rodents. Rodents have a short lifespan, so very often the neurodegenerative part of the phenotype is hard to produce, or you find that the mice do not quite show the behavioral phenotype that you see in humans. So we actually generated 2 mouse models, and I don't want to bore you with details, but it's important to know that these are technologies we have at the moment when you read about animal models. Our first model is really our workhorse. We targeted expression of a mutant cDNA, so a part of the human gene without introns, just the exons put together, and we targeted it to Purkinje cells. So this is a good model. I mean, Purkinje cells are lost in SCA2, but there's clearly, as I showed you, there's much more to the phenotype than just cerebellar disease. So that model is good to look at the cerebellum, but would not apply to other neurons. And then more recently, we were able to put the entire human gene into what is called a bacterial artificial chromosome, inserted that in the mouse, and so we now have a mouse that expresses the entire human gene, including regulatory regions and exons and introns. And as you might expect in that mouse model, we actually see expression of the mutant protein in deep cerebellar nuclei, in brainstem nuclei, and especially also in motor neurons. So how do you measure the behavior? And it's really not as easy as you would think. You can't ask a mouse to do a paw-snout maneuver or do rapid alternating movements. So most of us have used this machine that's also very well known in the ALS field, the rotating rotor rod. You put mice on this rotating rod. It accelerates, goes from 0 to 40 or 50 RPM, and you measure how long the mice stay on it. Sounds easy, but when you deal with mice a lot, you find out some mice are very curious. They just turn around, want to run backwards just for the fun of it. And so you get actually quite significant standard deviations. You can also do a beam walk, similar to tandem gait analysis, or you can do video analysis. In our hands, none of these more advanced methods have really worked. Of course, one thing you can do in mouse models that you can't do in humans is you can sacrifice mice at different stages and ask, well, what has changed? And so you can look at the morphology, you can count Purkinje cells, you can stain their dendrites with calbindin, an antibody to a calcium-binding protein that shows up the dendrites very beautifully. Or what I will briefly touch on, you can also nowadays just sequence all the transcripts that are present in the cerebellum, called the transcriptome, and look at disease progression from that point. So this is what we found when we used one of our mouse models in black up on top. You see the wild-type mice. They learn this task being on the rotor rod easily, and up to several months old, they perform this task very well. On the other hand, when you look at the transgenic mice, at 4 weeks, they perform just like the regular mice. So it tells you that the cerebellum is laid down correctly, their behavior is normal, and then they show a progressive decline in motor behavior. So it models, recapitulates important parts of the human disease, that initially you're normal, and you have a later onset, and then that onset is progressive. So what about morphology? And that's actually an important slide, and it's replicated in many other models. And the take-home message here that cell death is actually a relatively late event. And that gives us all hope in the clinic, because we see patients usually not pre-symptomatically. We see them when they have some symptoms. And there's still nerve cells left that are dysfunctional, but that could potentially still be rescued. So what you see here in the panel on the left side, on the top is staining of Purkinje cells and the beautiful dendritic arbor with a calcium-binding antibody. You can see the Purkinje cells nicely lined up. I can use this here. Yes, so here are the Purkinje cells. Here are the dendrites. Here are the transgenic animals at 4 weeks, still nice Purkinje cells, dendrites, and then you immediately see that Purkinje cells are starting to drop out very late, and now the dendrites have shrunken. For those of you who like aggregates, there are also aggregates in this mouse model. But when you plot the morphological changes, remember at 8 weeks, that's where we started to see behavioral changes. The molecular layer, the dendrites are still normal, and Purkinje cells drop out relatively late by 36 to 40 weeks. So there is a long prodromal stage where mice are symptomatic, but there's no cell death. With Tom Otis at UCLA, we started also to look at the function of Purkinje cells. We prepared cerebellar slices. So you actually take an animal, slice the cerebellum, put it in a culture dish, and then you extracellularly record from these Purkinje cells. Purkinje cells are spontaneously bursting cells. As you can see here, fire rate at about 40 hertz. In the wild types, they do this until the cows come home, until 1 or 2 years. In the transgenic animals, again, you see they're normal initially, and you don't have to be a physiologist to realize that they start to fire slower and slower, but still regularly. When you plot this, and again, what is important, 8 weeks was the symptom onset on the rotarod. That's when the Purkinje cell firing frequency for the first time becomes abnormal. Well, if you think about developing biomarkers or the earliest signs of disease, this is a way to get at this. We started to prepare RNAs from mice at different ages, and we found that already at day 1, a significant number of messenger RNAs were dysregulated. And that dysregulation, or the number of dysregulated genes, increased with time. And they had very different time courses. Here is a particular transcript that is already abnormal early on and then rapidly declines. Here's another one that is the inositol triphosphate receptor that is initially normal and then declines. And potentially down the line, these may become biomarkers to measure the progression of the disease. So to summarize what I just showed you, if the black line here tells you about the motor performance, the transcriptome dysregulation starts early. Just at the time when the behavior changes on the rotarod, you see changes in Purkinje cell firing that are progressive. Much later, at 16 weeks, you see dendritic changes. And again, much later, at around 30 weeks, you start to see dropout of Purkinje cells. So how do you get to therapies? And for those of you who are old enough in the room, I think in the 90s when we were starting to find diseases, still in very laborious times, when you look back, even I think this was like going on a sea voyage and having no maps. That's really what it was like. We had no maps of the genome. We had a few markers like islands. Nowadays, you just sequence the whole genome pretty straightforward. The young generation has no appreciation how difficult it was, but that's life. But we all thought we find the gene, we find the protein, we somehow know what it does. And if we don't know what it does directly, we do interaction studies. We see what other proteins interact with our disease protein, and then we'll stumble onto a pathway. We'll modify the pathway, and we have treatments for Huntington's disease, or ataxias, or Alzheimer's disease. Clearly, that was not the case. And really, one of the problems was that you find a large number of proteins that interact, and you don't quite know which one now is really the important one. And you do a lot of these interaction studies in convenient cell lines, and you don't quite know if this interaction is relevant in your cell of interest, be it motor neuron or Purkinje cell. And they're actually hard to culture and manipulate. And for those of you who work with motor neurons, you know that the phenotype in the dish is very often an embryonic motor neuron phenotype. It's not the adult motor neuron phenotype. So it's complex. But we engaged in this, and we and others found a number of proteins that interacted. But it did not result in a therapy for SC2 and for other diseases. So in 2011, we took a very different approach. We actually wondered, can we target the gene directly? And there were two basic questions, actually. Can we target the gene? And the second one, is it safe to do this? And it was clear to us that in our approach, we would have to target both the mutant allele and the wild-type allele, so we would reduce expression of ataxin-2. Well, fortunately, we could answer the second question relatively easily, because we had actually generated a knockout mouse of ataxin-2. So we had generated mice that either lacked one copy of the mouse ataxin-2 gene or both copies. What is interesting, they become very, very obese. I've never gotten funding for this from the NIH, despite I just said they have been good to me, but the obesity mafia didn't want yet another mouse model of obesity. It's actually very interesting. If you give them fat, the classic American diet, they really blow up. They're more obese than leptin mice, or leptin receptor mice. But that is one story, but we were clearly interested in cerebellum and CNS. Is it normal? Is it safe to be without ataxin-2? So we looked at morphology, the physiology, and transcriptomes. And here's what we found. When we looked at morphology, here is a normal mouse, Purkinje cells, stained with an antibody to ataxin-2, nice staining. Here are the dendrites. Here is a knockout mouse, no staining as you would expect, but you can see the Purkinje cells are here. The molecular layer is same thickness as in the wild-type mice. We also looked at physiology, and in the slice, the Purkinje cells fired away happily at around 35 to 40 hertz. And when we looked at transcriptomes, in contrast to transgenic mice where we had a lot of changes, the knockout mice had very few. So, so one thing was we could conclude that by very detailed methods, lack of ataxin-2 was fine with normal cerebellar functioning. This was also the ultimate proof that SC2 is not caused by loss of function, because if it had been, we would replicate the same phenotypes that we had seen in our transgenic mice. And we didn't. There was normal function, normal morphology, and even at the transcriptome level, there were no changes. So that gave us a lot of confidence that going forward, trying to reduce ataxin-2 would work. So how did you do this? And there was a chance occurrence in 2011 at Society for Neuroscience. I ran into an old collaborator of mine who had moved to Ionis, and I said to him, you know, Gene, you now work for a company that makes these new compounds, Antisense, that can target genes. Would Ionis be interested? And I'd actually no hope that they would be interested in a rare disease, but it was only 7 years ago. Nobody knew that this strategy would actually really work. They were very interested. And they started to screen antisense oligonucleotides for us against ataxin-2. So how do these antisense oligonucleotides work? I will have another slide in a second to show you. These are not your grandfather's or grandmother's antisense oligonucleotides that some in this room may have used 20, 25 years ago. They're highly modified. So what they do is they find the DNA molecules, they find their target RNA. Now an RNA-DNA hybrid is formed. There's an enzyme that all cells have called RNase H that detects these hybrids, because they're very often caused by foreign invading DNA, destroys the mRNA, but leaves the antisense oligonucleotide intact, so then this ASO can find another target and re-engage in target destruction. And that actually works. And these modified ASOs stick around in the body for months at a time. So actually, with a relatively small screen of 152 ASOs, we identified 15 that in cell models reduced ataxin-2, and then Dan Scholes took this on in my group and put them into mice to see if it also would reduce ataxin-2 in vivo. This slide just shows you the phosphate backbone of a DNA base and the sugar molecule, and you can modify the phosphate backbone, but you can also modify the sugar. These are called Mo oligonucleotides. And then you can mix and match different modifications. And when you do this, it's called gapmer. And these ASOs then require very good properties for therapy. They are tolerable. They stick around in the CNS for a long time. They are specific and stable. And the stability was a big issue when we initially tried it. And they're very good at recruiting this enzyme RNase H to destroy your target mRNA. So I think takeaway from this slide is that you can use ASOs like any small molecule now to affect splicing. For those of you who do neuromuscular disease, you know nusinersen is FDA approved now, where you actually don't destroy an mRNA, but you change its splicing pattern. You can use it most commonly to destroy a mutant RNA, but you can also potentially use it to increase expression of an RNA. It just depends how you structure the ASO and where it binds. The one disadvantage with ASOs is at the moment that they do not cross the blood-brain barrier. So you have to inject it into the CSF space. Dan Scholes became a master mouse neurosurgeon and learned how to inject the ASOs into the lateral ventricle. That is clearly a disadvantage, but it is also an advantage because you don't have to worry that much about systemic side effects. I mean, you don't have to worry about your ASO causing some cancer in liver or causing cardiac arrhythmias. So, and then potentially another advantage is that in contrast to current viral therapy, the action is long-lasting, but it terminates. Whereas with viral therapy, where the virus integrates, you are stuck with that integration event and whatever this virus does for the rest of your life. So when you inject these ASOs in the ventricle, you can stain the ASO backbone with a nice antibody, and you see it's expressed in Purkinje cells, also in deep cerebellar nuclei. I'm not showing you here. It's also very heavily expressed, well, taken up by motor neurons. This is an active process. Not every cell type in the CNS takes up ASOs as avidly as Purkinje cells or motor neurons. The effect lasts a long time. When Dan injected an ASO here, we could see reduction of ataxin to up to 70 days and longer. So we then started to do preclinical mouse trials. And who here in this room has done clinical trials in humans? I'm just curious. Well, a fair number. So you all know how we do it, right? That is not always the case how we do it in mice. And I just want to point out certain things that we thought was good practice. So we used symptomatic mice and then randomized them into two groups. We also used a mixed mouse background. If there are any mouse geneticists here, generally people like to use highly inbred lines. That is as if we were doing clinical trials in young college students from UW. Clearly, it doesn't generalize to the other sex or to older age groups. And we did all our testing blinded. We had a primary outcome that was the rotor rod performance, and we had a secondary outcome that was a reduction of ataxin-2 at the end of the trial. That's actually pretty much how the trials with ASOs are now done in humans, of course, and how clinical trials are done. So this is what we found. We injected, another thing we did, we decided to really put it to the therapy to the test by using symptomatic animals, not pre-symptomatic. Most mouse studies are actually done pre-symptomatically, which is cheating to some degree. So we injected them at 8 weeks when they had already a rotor rod deficit, and then measured their performance on the rotor rod. In red here, you see saline injected mice, and as you would expect, they progress in that time period. Here, the ASO injected mice, and they stabilize in their rotor rod performance. At the end of the trial, we sacrificed the mice, and we looked at expression of human ataxin-2 in the cerebellum, saline injected mice, here the ASO injected mice, and even 10 weeks after injection, they had a 75% decrease. So our secondary outcome criterion was met as well. Well, for those of you who do mouse studies, it's not that different from dealing with humans. The behavior is very variable. I don't know if there are any runners in the room, but some mice are runners, others are couch potatoes, and there's very little you can do about it. You just have to have very large numbers. So that's why I had mentioned to you that we are looking for other markers of success. And we can both use transcriptomes and proteomes, and I will only show you the effect in the acute cerebellar slice. And this is what we saw. When we treated mice with saline, we saw the expected decrease in firing frequency. When we looked at mice at the end of the antisense treatment, their Purkinje cell firing frequency had gone up to the wild-type frequency of about 40 hertz. So that was a nice independent outcome marker that showed us that the antisense therapy was working. So to summarize this part of the talk, and this applies to a large number of ASOs, they have a wide CNS distribution after interventricular injection all the way down to the spinal cord. They have a long-lasting effect, days to months. And we showed actually these studies that I just showed you in 2 different mouse models. I had already mentioned to you, and I think it's important, this is a room, I think, full of clinicians, and it's important to recognize the important contributions that clinicians can make with their observations to understanding of disease and to therapy. I'd already mentioned to you that in addition to ataxia, a lot of SCA2 patients have brisk reflexes, then they start to lose their reflexes, they get weak, some of them have fast circulation. So you're already getting the point, could be an ALS-like phenotype. At the same time, some patients have rigidity, slowness in initiation of motor tasks, and they respond to L-DOPA, so they look a little bit like Parkinson's disease. And indeed, since we identified the gene in 1996, pedigrees have been described that have L-DOPA responsive Parkinson's disease, seen by movement disorder specialists, not by a person like me who does ataxia, but real Parkinson's and they have garden variety Parkinson's disease, but underlying they have an SCA2 mutation. The same is true for ALS, and there are now some pedigrees described that have ALS in some members and SCA2 in others. So that was an important clinical recognition, but it didn't really take hold until a molecular scientist at Stanford, Aaron Gittler, who's a yeast and fly geneticist, asked the question, what does the taxon 2 interact with? And he pulled out a very interesting protein called TDP-43, which is intimately involved in ALS and in frontal temporal lobar dementia. So when he showed that ataxin 2 and TDP-43 aggregate together in a structure called a stress granule. Who has heard of stress granules here in the audience? I'm just curious. It's not surprising. It has entered neuroscience actually very recently. It's an important way of a cell, initially discovered in yeast, to deal with stress. So if you encounter stress, nutrient stress, for example, temperature stress, the last thing you want to do is spend a lot of energy on making RNAs into proteins. So what does the cell do? It takes all these untranslated mRNAs, puts them in a stress granule, prevents them from translation, waits until the stress is over, releases the mRNAs, and the yeast cell lives happily ever after. This was discovered a long time ago, and recently has been found that this is a very important pathway that goes wrong in neurodegenerative diseases. It's probably one of the hottest areas of neuroscience at the moment. So ataxin 2 is actually involved in the formation of stress granules together with TDP-43. And what Aaron did is he found this, then he knocked ataxin 2 down in the fly, and he found that the flies, he could restore neurodegeneration caused by mutant TDP-43. He also then started to look at the structure of ataxin ST2 repeats in ALS patients. And he found that when you had more than 27, your risk for ALS went up. And being a geneticist, I think one thing you learn is that the precision of allele length is very important. So actually, let me just back up 1 second. This slide shows you that TDP-43 is important in ALS way beyond its mutational range. So mutations in TDP-43 itself are relatively rare in ALS, but most ALS patients have TDP-43 aggregates. So it makes it the stress granule, the TDP-43 granule is an important thing to target. And the same is also true for FTLD. A lot of patients who do not have direct mutations in TDP-43 may form aggregates. So let me come back to the role of ataxin-2 repeats in ALS. And we are talking not about pathological repeats. You may remember that 33 and more gives you ataxia. We are talking about repeats from 27 to 33. And the question was, if you have these long normal repeats, do you have an increased risk of ALS? So a student in the lab, Annelies Neuenschwander, did a meta-analysis of all the published studies. And this is what she found. When we compare presence of certain allele lengths in normal individuals or in ALS patients shown in dark, up to about 28, there is no change in the relative risk. And at 30, it starts to increase. And actually, by 32, it has reached an 11-fold increase. So this is a significant risk factor for ALS. Fortunately, these type of repeats of 32 length are relatively rare in the population. So you can think about it as if this were a very potent toxin, but fortunately very rare. This is what this genetic risk does. But as a geneticist, I think when we counsel patients, it is important to know what the risk is for 29 repeats versus 32, because you don't want to tell a patient, well, you know, your repeats are over 27, you have a 10-fold increased risk of ALS, because we now know that 27, 28, and 29 repeats have normal risk. This has been replicated by other meta-analyses, so it's an important link between ataxin-2 and ALS on a clinical and risk basis, not a Mendelian deterministic allele. So, we then collaborated with Aaron and asked the question in the fly, reduction of ataxin-2 worked. Would it also work in mouse models? And Aaron used a particular model of ALS that overexpresses wild-type TDP-43. It does it 100-fold to normal, so you worry a little bit, how much does it replicate ALS? It also leads to very early death. Mice die within the first 30 days. So again, I just told you that we ideally like to have mice be normal into early adulthood and then show degenerative disease. The way we looked at targeting ataxin-2 was in two ways. We used genetic interaction. We used our ataxin-2 knockout mice to reduce ataxin-2 expression, and then Aaron used ASOs. And this is what we found. So when we here is the survival of these TDP-43 mice, and you can see by day 30, there's not a single mouse alive. When we reduce ataxin-2 by 15% by breeding with ataxin-2 mice, we prolong survival. If we knock ataxin-2 down or out completely, we not only have a prolongation of median survival, but for the first time, we have long-term survival, something that you just don't see in this mouse model. When you use antisense oligonucleotides, you see a similar effect. The reduction was 75%, and you can see how dosage sensitive ataxin-2 is. With 75% knockdown, you get an intermediate result between 50 and 100, which gave us rise to with Ionis to collaborate to find more potent ASOs that really knock ataxin down to probably 5% of its normal level. So ASOs are making it into clinical practice. Nusinersen had mentioned is already approved for SMA. Eteplirsen is FDA approved for Duchenne muscular dystrophy. The phase 1 trial for Huntington's disease was just finished. SOD1 trial is ongoing. There are mouse models of tauopathies where ASOs against tau appear to work. We and Aaron Gittler have worked on SC2 and ALS, and Hank Paulson at Michigan is working on ataxin-3, and there are others, other mouse models. There are even, I'm aware of, one single patient trial in Boston where the FDA has allowed to synthesize an ASO for just a particular patient and use it as a last-ditch effort. Before I leave this, I think we're all clinicians in this room, and we have all heard about drugs that work wonderfully. And I think we have to take the same critical approach to ASOs as we're taking to drugs. These have effects, they may have side effects, and we have to test them very critically. It makes a lot of sense. ASOs have one thing that is mechanistically very nice. They target a gene that is mutated, that causes disease, but it still can mean that they can have off-target effects. And we discussed this last night at dinner. Some ASOs also activate glia and microglia. So we really, the proof is in the pudding. It's an empirical test, not a philosophical test. So the last few minutes I will want to spend on testing a little bit. I want to give you some highlights how I think about testing, especially testing in the ataxias. So why even do genetic tests? Most of these diseases are really incurable. Anybody here in this room who deals with chronic neurodegenerative disease knows it. But I think we also all know that our patients would like to have a name for a disease. I think that it's incredibly important to know what you are facing. Actually, when you find a genetic diagnosis, it actually ends a lot of unnecessary testing. I mean, we have now reached the point where it is easier to order a very extensive gene panel than doing the MRI every year. When Mrs. Smith comes to me and I say, let's do an MRI. $1,000 out the window. Have I learned anything? Probably not. Cerebellum maybe has gotten a little bit worse. Maybe the brainstem has shrunk a little bit. It really doesn't help you or the patient, right? The exam is a lot more important for the patient. Can she still walk, go to the bathroom without falling? It allows reproductive planning. Friedreich's ataxia is a recessive disease. SC is a dominant, very different risk to offspring. It may help you in prognosis. We now know that SC1, SC2 progress relatively fast. SC6 progresses so slowly that we will not be able to conduct clinical trials because you cannot measure any progression in a reasonable fashion. You can try to avoid cerebellar toxins, alcohol, lithium, galantin. With reproductive choices, we can clearly prevent the disease, and we are now entering, this slide is, I made this a long time ago. It was before we even thought about disease-specific therapies. We are now there, and I think within 2 years we will see targeted therapies for SC2, just as we're seeing for Huntington's disease now. So what do you get when you see patients with late onset cerebellar degeneration? You can see this is a relatively old study, but it is still true. This was a study done by Laura Ranham at Minnesota, who is now in Gainesville. They took 3 classes of cerebellar patients. Patients who clearly had a dominant inheritance, so one of their parents was affected, recessive, where one or two siblings had the disease, but the parents were normal, and they excluded the typical Friedreich's phenotype, so that's very important. So age of onset after age 25, upgrowing toes, dropped reflexes, kyphosis, the usual stuff for Friedreich's. And then took sporadic patients who were singletons and had no parents involved. This is what they found, and this is actually really still true today, 20 years later. So clearly if you test for the most common SCAs, you have about a 2/3 take rate and find mutations. When you look at recessive patients, or seemingly recessive, you find Friedreich's. So that's the first important take-home message. Friedreich's ataxia can start late. We now know that about 25% of patients start after age 25, and they actually have a slightly different presentation from the typical childhood onset patients. But then there were also SC2 and SC6 mutations. We now know why this is, because these CAGs repeats can expand from parents to offspring, so it's possible that offspring have a disease before the parents manifest it. But also surprisingly, some sporadic patients, actually 10%, had dominant SCA mutations. Why is that? Well, there's adoption, there is non-paternity, and again, there's the same thing. The parent may have a repeat of 29 repeats that then in meiosis expands into the pathological range, leading to seemingly sporadic presentation. I think this is important because it makes the point that even in sporadic patients when the family history is not deep and accessible, it is useful to test. So I will run a case by you. It's actually very simple, but genetics will make it complicated, at least for a while. So this is a 55-year-old man of Ashkenazi extraction, progressive unsteadiness for 3 years. For 1 year is mild erectile failure. Family history, father died of Parkinson's, mother is still alive and well. He has 3 siblings, all alive and well, and he has 3 children in their teens. So just think about what you would think this could be. Well, then you look at his MRI. Cerebellum is small. Actually, if you look at the brainstem, I would say that the pons doesn't look normal to me. It was not called out. Actually, by the way, very typical for many radiologists don't even detect this. I'm not talking about neuroradiologists, general neurologists. So in my practice, I insist on seeing the MRI because the report is not reliable. And then when you look at his pons here, you can probably see something. Anybody wants to venture a guess if this walked into your practice, what you would think this is? Multiple cysts. It's an MSA, right? I mean, 55 years old. He's right. I didn't make this up. This is exactly the median onset of MSA. He has early autonomic failure. He has the hot cross bun sign, or I, as a non-religious person, call it the plus sign. And he has cerebellar atrophy and pontine atrophy. Pretty straightforward, right? No family history. MSA is, as far as we know, not a genetic disease. So let me advance. So what happened? Patient had this is a very wealthy patient, had a primary care physician who thought, well, this is ataxia, let's do a panel. This is a bit older. This was an Athena panel, but it would apply to any panel you do today. And he found a mutation in a potassium channel. This was a serine to glycine substitution. They called me because my group had identified this gene. It causes SC13. Patient was absolutely convinced he had SC13, which is a very rare autosomal dominant form of spinocerebellar ataxia. And what to do now? So this is not that uncommon in your practice. When you do testing, you will get variants back, and you will have to ask yourself, well, is this meaningful? Does this explain what the patient has? So these are the thoughts I go through. Is this phenotype typical for SC13? You can do a PubMed search. Many of you may know about gene reviews. It was actually started here in Seattle at the University of Washington. It was it still is an online textbook. The site, unfortunately, is down now, but you can find it through PubMed and putting SC13 and gene reviews in, and you find a very up-to-date, very practically minded explanation of what SC13 is and what mutations you see. Then you should ask yourself, if this is really a causative mutation, is the DNA change found in normals? Who else in the family carries this variant or mutation? And is there an effect a predictable effect on the protein function? So let me answer this for you. SC13, as we described it, there are 2 forms. There is an early onset phenotype with ataxia, mild ataxia, and cognitive impairment. And then there's a late onset, almost pure cerebellar ataxia that is very slowly progressive, no autonomic failure, and it is highly penetrant. Whoever has a mutation will develop the disease. So, is the phenotype typical for SC13? No. Is the DNA change found in other normals in the family? Yes. And actually, I think one of the sisters and 2 of the kids carried that mutation. I excused myself from the case because it became so difficult, but I think it is almost criminal to tell the kids that they have inherited a mutation that might cause a disease like their father has. And what is the likely effect on the protein function? It's benign. Let me show a little. So my diagnosis was MSA, and indeed I've called the patient up. He has rapidly progressed. This is multiple system atrophy. None of his family members have become sick. So if this is a mutation, you can ask yourself, well, does it occur in the normal population? We now have databases where you can look at 50 to 100,000 people, and you can ask yourself, how often does this change occur. And actually, it occurs in 9 out of, 12,000 people. So it is rare, but not unheard of. So this is a rare normal variant. You can also go on, and there are many programs that actually companies, when you order panels, they use all these programs that try to predict what the effect of the variant is on protein function. These are all nice names, from mutation taster to polyphen, and the prediction for this variant is that it has benign or only moderate effects. So everything points to this point that this patient has MSA, and the variant that we found is an associated benign variant. This happens every day in clinic, and in my experience, the hardest thing is when you are the physician and you say, well, I don't think you have SC13, Mr. Jones, and Mr. Jones gets very upset because you just took the disease away from him. And that's something we will see more and more the more we test. So next generation sequencing, which really most companies do nowadays. Even if you order a gene panel, let's say you order an ataxia panel from the University of Chicago, they do an ever-growing number of 300 to 400, 500 genes. They actually don't test each gene. They do whole exome sequencing and then just report these 300 genes out that are of interest to you. What you have to know is that you cannot detect repeat disorders at the current state of technology. So if you think somebody had SC2, your whole exome or your whole genome will likely be reported as normal. Same for Huntington's disease. That's an important take-home message. At our current state of DNA sequencing, it will likely change in the next years. You may still miss many smaller insertions and deletions. You will miss complex rearrangements. You miss if they are what is called homopolymeric tracks. When you have long tracks of A's or C's or G's, it's still hard to align. You have to make sure that in whole exome sequencing, some exons of genes are not covered well. So you may have an average coverage of 30x, which should be enough to detect mutations. In your gene of interest, just by chance, the coverage may drop down to 2x, and then you don't find the mutation. So we actually know if we test clearly genetic diseases, we only find about 50% at the moment. So the take-home message here is not Mr. Jones, your whole exome sequencing was normal, you don't have a genetic disease. The correct answer is you have a genetic disease, but we have not been able to find the mutation yet. So for those, I know nobody came late, but this is one of my favorite slides to summarize it. And before I do this, just want to acknowledge a large number of collaborators. This is a part of my lab that worked on the ST2 project. Tom Otis was chair of neurology at UCLA. He did the neurophysiology, and these are our collaborators at Ionis. And so what I wanted to show you in the first part of the talk that antisense oligonucleotides have now made it into the clinic. They are completely novel compounds that allow you to target genes specifically and for a long time, but in contrast to gene therapy, not forever. So there is a time of about 6 months when the effect will wear off. And I think in the second half of the talk, I wanted to give you a glimpse at some of the complexities of next generation sequencing. And this is a city that likes food. And when I wrote this last night, you have to be like a foodie in a good restaurant. You have to keep your critical approach. And true foodies are always critical. The steak is never quite right, and the pizza too well done. So that's the way you have to approach variants, with a critical mind. Thank you.